Hello everyone. Today we're going to be looking at the basic histology of the glomerulus. So the kidneys, one of the most important vital structures in human body. They are composed of millions of individual structural and functional units called the nephrons. Each nephron is composed of two structures, the glomerulus and the tubules. So the glomerulus, you might find the term renal corpuscle or Malpighian corpuscle. Those are other names to term glomerulus. So the Malpighian corpuscle is named after an Italian anatomist and histologist called Marcello Malpighi. So glomerulus has two structures in it. The first one is the glomerular capillary, and the second one is the Bowman's capsule. So the capillary is a branching network of arterial lupus, whereas the Bowman's capsule is the sac-like structure upon which the glomerular capillary rests and embedded in where the ultrafiltrate of the glomerulus enters into it. So the Bowman's capsule has a potential space within it called the Bowman's space, or sometimes called the urinary space. So when we look close up, the glomerular capillary is made up of lining cells called endothelial cells. These are flat or squamoid, which means flat cells, and having a membrane fenestra, which is small pore-like openings found on the surface membrane. So their function is it allows the plasma fluid to enter the cells and exit into the Bowman's space. So these cells, endothelial cells, they have oval to round nucleus, which is centrally located, and they are light pinkish when you stain them using the eosine staining. They stain light, which means light pink cytoplasm, and they have oval nuclei, giving them their characteristic appearance. So each endothelial cell lies upon a basement membrane. So this basement membrane is called the glomerular basement membrane. What is this structure? Let's see it. So this structure is a trilaminar structure, which means it has three layers and it functions as a filtration barrier, which means the blood which is in the capillary cannot exit the capillary wall and enter into the Bowman's space, because if it happened it would be pathological, that is hematuria. So it is prevented by the protective effect of this membrane called the glomerular basement membrane. So it has three ultra microscopic structures, which means those structures can be appreciated by using electron microscope. So we have the central electron dense layer called the lamina densa, and two thin rims, one found within the capillary lumen is called the lamina rara interna, and the one which exposed to the urinary or the Bowman's space is called the lamina rara externa. So it has a thickness around 345 nanometers, which is very, very minute. So if you ask what are the composition of this basement membrane, what made up this layer? If you ask this one, it is composed of collagen, particularly collagen 4, laminin, and one important structure I want you to remember is the intactin. What is this intactin? This is a transmembrane protein which helps to attach the food process of the podocytes to the basement membrane. We'll see them later. So this loss of intactin, or sometimes it is called nidogene, it helps to firmly adhere the food process of the podocytes to the external layer of the glomerular basement membrane. So the next cellular populations are called the podocytes. They are also called the visceral epithelial cells. These cells, along with the parietal cells, they constitute two of these cells, the visceral epithelial cells or the podocytes, and the parietal epithelial cells. Together they compose the Bowman's capsule. So the space between those two cells, this yellowish area, is called the urinary or Bowman's space. So the podocytes, they are regarded as the largest cells of the glomerulus. So they have this large centrally located nuclei called the cell body, and cytoplasmic processes which arise from the center called food processes. So the initial or the first food process that arises from the cytoplasm is called the primary, whereas a secondary branch which arises from the primary food process is called secondary food process, termed as pedicle. So the function of this pedicle is they interdigitate and intertwine together with the adjacent another podocyte food process, forming a glomerular basement membrane-like structure called filtration slit diaphragm. So this structure acts like a glomerular basement membrane. It strengthens, it further makes the glomerular basement membrane more strong, more firm, preventing leakage of important plasma proteins. As we've said earlier, each processes, the food processes of the podocytes, which rests upon the basement membrane, are attached by important proteins called the nephrins. So these nephrins are an important transmembrane that attaches the food process firmly to the basement membrane. So the loss of protein of these structures will result in nephrotic syndrome. So subsequently, protein will be lost through the urine. So these parietal epithelial cells, they are flat, squamoid, containing oval to round centrally located nucleus, and they have a pale eosinophilic staining cytoplasm. So their main function of one of the main functions of the parietal cells is, one, they prevent the ultrafiltrate of the plasma from leaking into the surrounding periglomerular tissue, right? So they prevent, they act like a barrier. Second, they have a proliferative potential, which means during an injury, the parietal epithelial cells, they can renew, regenerate themselves to transdifferentiate, giving another population of podocytes, which is one of their important functions. So they have a proliferative potential. The last and important cell groups are called the mesangial cells. So it comes from the Greek term mesos, which means between, and angion, which means a vessel. So this means mesangial cells are cells which are situated between two adjacent capillary loops. So they give structural rigidity and support for the blood vessel, one, and second, they regulate the glomerular filtration. Why? Because when you close-uply look at those cells, they contain actin and myosin filaments, which have contractile abilities. So when these mesangials contract and relax, they adjust the caliber of the capillary lumen, hence regulating the glomerular filtration. And so the third function is those cells, they phagocytose or eliminate and degrade unimportant proteins and immune complexes. And lastly, they can elaborate important cytokines, prostaglandins, hence by regulating the kidney function. And that is it.